Endometriosis and PCOS are the yin and yang of reproductive medicine. Endometriosis is linked to relatively low levels of prenatal and postnatal testosterone. At low levels, testosterone alters the trajectory of reproductive and physiological development, predisposing to endometriosis. By contrast, PCOS is driven primarily by higher prenatal and postnatal testosterone, resulting in an opposite set of traits. The hypothalamic-pituitary-ovarian axis, or HPO axis, is the command system that regulates reproductive function. It begins in the hypothalamus, which releases gonadotropin-releasing hormone, or GnRH, in a pulsatile rhythm. GnRH stimulates the pituitary to release follicle-stimulating hormone, FSH, and luteinizing hormone, LH. FSH promotes follicle growth and maturation, while encouraging estrogen production from granulosa cells. LH triggers ovulation, releasing a mature egg, and transforms the follicle into the corpus luteum. The corpus luteum secretes progesterone and estrogen to prepare the uterus for pregnancy. Estrogen provides feedback to regulate GnRH, FSH, and LH, maintaining cyclical harmony. This feedback loop sustains early pregnancy if conception occurs, or leads to menstruation if it does not. Prenatal testosterone is present in females too, produced by fetal and maternal glands, ovaries, and fat tissue. Maternal testosterone also crosses the placenta, influencing the fetus's hormonal environment. Prenatal testosterone levels can be tracked by anatomical markers, such as anogenital distance. Another marker is the ratio of the second to fourth finger, the 2D4D digit ratio. In women with endometriosis, shorter AGD and longer 2D4D ratios reflect lower prenatal testosterone exposure. Endometriosis is marked by hormonal patterns: lower LH, lower testosterone, higher SHBG, higher oxytocin, and others. Epigenetic changes drive the abnormal survival and implantation of endometrial tissue outside the uterus. Hypermethylation of progesterone receptor genes contributes to progesterone resistance, a hallmark of endometriosis. Histone modifications alter chromatin and gene accessibility, fueling inflammation and angiogenesis. Dysregulated microRNAs further disturb immune response, cell migration, and invasion. In PCOS, epigenetic changes affect genes regulating androgen production, insulin sensitivity, and inflammation. Hypomethylation of androgen-producing genes like CYP17. A1 drives hyperandrogenism, leading to acne, hirsutism, and metabolic issues. Histone changes alter chromatin accessibility, affecting follicle development and insulin sensitivity. MicroRNAs in PCOS tissue show different binding patterns compared to normal tissue, disrupting gene networks essential for proper follicle growth, inflammation control, and insulin regulation. The disrupted networks appear as broken or misfiring pathways, with inflamed tissue glowing red. Dysregulated microRNAs disturb gene networks for follicle growth, inflammation, and insulin regulation. Despite their opposite presentations, both show similar patterns of epigenetic dysregulation, highlighting their common mechanistic foundation. Both disorders arise from epigenetic dysregulation, though they manifest as opposite reproductive syndromes. By understanding these reversible epigenetic changes, we open the door to therapies that restore balance and improve reproductive health. But reproductive health is only part of the story. In our final episode, we'll explore how heritable epigenetic changes can shape the developing brain itself, influencing conditions like autism spectrum disorder, and what this means for future generations.